everything we've considered so far has seemed like bad news for formation of the double helix structure. So we saw that entropy opposed it. This tendency of energy to become dispersed has the consequence that it favours structures where the chains are not organised in a specific way like this. And then when we thought about the folding of the backbone, we saw that the additional proximity of the negatively charged phosphate groups means that electrostatically this structure doesn't look like it should be favourable. So there has to be something going on which drives the formation, because we know that this is what DNA does look like. And so what we haven't looked at so far are the bases. So that's what we're going to have to do now. Now, when you look at this kind of representation of the DNA structure, or indeed if you look at the familiar sort of ball and stick representation, you get the impression that the base pairs are sort of floating around in the middle of the structure with sort of big gaps in between them, which you can see sort of, if we stop this spinning round for the moment, you see these sort of big gaps in between the bases, and you have the feeling that they're sort of therefore floating around in empty space, except that of course we know it's not empty space, it's dissolved in water, and you might be forgiven for thinking that there would be hydrating water molecules all the way around the bases. However, if we change the representation and make a space-filling model where we replace these artificial sort of lines in between the atoms which represent bonds, and recognise that in reality, of course, the atoms squash up together as closely as possible when they form bonds. If we build a space-filling model for the bases like that, it looks like this. So here you can see the backbone curled around the outside and the bases, as we know, lined up down the middle. But the big revelation here is that there are no gaps between them. They are in close contact all the way down through the middle. So it's not a sort of damp core structure. It completely excludes water because the bases are piled on top of one another, and because they're aromatic and therefore flat, they will form a very densely packed stack like that. So what we need to do is to understand what's going on in that core structure so that we can see if that will give us the clue to how this double helix folds. So let's remind ourselves what these bases look like. And just looking at the skeletal formulae, the striking thing is just how polar this range of atoms around the outside is. So all these nitrogens and the oxygens are a lot more electronegative than the carbons they're bonded to. So we've got a great deal of polarity in these atoms around the fringes of the rings. And what we can see in this picture is that this creates a great deal of potential for forming hydrogen bonds. So here we've sort of highlighted this. So the red dashed lines represent where we have delta positive hydrogens bonded to nitrogens, and therefore with the ability to form a hydrogen bond with something over here which is strongly delta negative, like an oxygen or a nitrogen. And then the blue lines represent the more electronegative atoms themselves, where they have the ability to form a hydrogen bond to a delta positive hydrogen on the other end. And the point about the picture is that there's a huge array of these hydrogen bonds potentially formable around the fringes of the bases. So what we need to do is think about the role that that hydrogen bonding could play in stabilizing the structure. And of course at this point you might feel that you're on familiar ground, because the hydrogen bonding between the bases is famous. in the double helical structure, and was indeed the key to Watson and Crick's original model. The way Watson and Crick put together their model is shrouded in controversy, but essentially what was known was the chemical structure of DNA, the arrangement of the bases and the backbone. That was well known, and it was also known from chemical evidence from a guy called Erwin Chargaff that the relative amounts of A and T in the structure are always the same, and of G and C in the structure are always the same. And so the idea that there must perhaps be some kind of pairing of G with C and A with T was known from chemical data. And then the key thing was that Rosalind Franklin and Maurice Wilkins, working in London, produced X-ray diffraction data, not from single crystals then, which give the atomic resolution we have now, but from fibres, which give lower resolution information. But Rosalind Franklin was able to get incredibly high quality diffraction data, which showed clearly, just from the overall shape of the diffraction pattern, that there was a helical structure here, and also revealed its dimensions. And that was what Watson and Crick sought to fit their model to. And the key thing that they did was to recognize this hydrogen bonding pattern between G and C and between A and T, which is central not just to creating a reasonable looking structure, but also, of course, to the way it works in biology. So here are the familiar Watson-Crick base pairs. So what they proposed was that in the structure, guanine always pairs with cytosine, and that this pairing is mediated by the fact that you can make this set of three more or less parallel hydrogen bonds between the two bases. Likewise, adenine always pairs with thymine, and again, you can make this time just a pair of parallel hydrogen bonds between the two. And it's not just the complementarity of the hydrogen bonds that was important for their model, but also the fact that if you look at the points of attachment, which are here and here, where these are the points where they're attached to the sugar rings, you can see that they're in essentially the same place relative to where the two bases are. And that's important for making the regular helical structure that we know we have to explain. So this is clearly going to be an important part of the structure, and we know from chemical analysis that indeed guanine is always paired with cytosine and adenine is always paired with thymine. So it all seems to fit beautifully together. And indeed, you will read in lots of books a statement that simply says that the double helix is held together by this hydrogen bonding. But when you stop and think about it, that's clearly, at best, an oversimplification. So we need to explore this in a bit more detail. So remember that our approach to this is to recognize that there is an equilibrium between the double helical structure, which we've been looking at, but also the potentially unfolded state. And so we can't just look at the hydrogen bonds in the folded structure and ignore what's going on in the unfolded state in order to assess the position of this equilibrium. And that's a very common flaw in the arguments that you read in textbooks and things, that they just talk about the hydrogen bonds here, but ignore what could happen if it unfolds. Okay, so let's approach this by starting with the folded double helix. So here we're looking down along the axis of the helix, so looking at it end on, if you like. So here are all the base pairs in the middle, and then around the outside, here is the backbone. And what the crystal structure also contains, but what we haven't really focused on so far, are water molecules. So let's see if we can have a look at them. And there they are. Okay, so those red blobs all represent water molecules which were present in the crystal structure when it was solved. Now, you don't necessarily get a very complete picture of hydration from a crystal structure because you're only going to see water molecules if they have fixed positions relative to the DNA. Remember that the whole principle of X-ray crystallography depends on all the molecules being arranged in a very regular way in the crystal. So if the water molecules have some freedom to move around to different positions as they do in liquid water, they may not show up here. The fact that there are so many of them, though, shows how important the water actually is and the structured water around the molecule is for stabilizing the double helix. So if we want to know how those waters are arranged relative to individual base pairs, it's quite difficult to see from this kind of picture. But what we can do is to take a slice. So if you imagine that, still looking at it from this angle, instead of looking at the whole cylinder, as it were, we're just going to take a thin slice through that cylinder. And there we are. So here you can see an individual base pair. And we've just sliced thinly through the structure, so there's a bit of the backbone there on the outside as well. And you can see there are a whole bunch of water molecules there. And we could move through the structure like that, just looking at different slices. So there's another base pair, again with a bunch of waters around it. And then here now we've added in the hydrogen atoms, which we don't normally show when we show crystal structures, but we can calculate where they would be, and so we can see those as the little white blobs here. So this is an AT base pair. Adenine is this double ring here, and this is thymine. And so what we have here are two hydrogen bonds directly between the base pairs, which is this delta negative nitrogen to this delta positive hydrogen there, and then this one between the NH2 hydrogen and a carbonyl over there. If we look at the waters, it doesn't show hydrogens on those because the computer doesn't know which direction the hydrogens of the water are pointing. But we can sort of infer what's going on by thinking about it. So when you have a carbonyl group, the oxygen will tend to form two hydrogen bonds, as it does in the liquid water structure, which you've learnt about in the chemistry course. And so you can see that what this carbonyl group does is, as well as forming a hydrogen bond to the NH over here, it forms another one to a water molecule here. Likewise, in the NH2 group here, this hydrogen is forming a hydrogen bond to the carbonyl group within the base pair, but this one is forming one to water. And you can see over this side as well, you've got a carbonyl group here forming a hydrogen bond to water. So that's the point, that the hydrogen bonding in the DNA structure is not only between one base pair and another, it's between the base pairs and water molecules. So it's then a fairly easy step to sort of say, well, what would happen if the double helix unfolded? Obviously, these hydrogen bonds between the base pairs would break, but they wouldn't be left high and dry, because all that would happen is that water molecules would rush in and take their place. So you can see the point is that either way, these bases get hydrogen bonding. In the folded state, it's a combination of hydrogen bonding within the DNA between the bases in the base pair and hydrogen bonding to surrounding water molecules. In the unfolded state, these hydrogen bonds within the DNA would be gone, but they'd just be replaced by different hydrogen bonds to water molecules, because the point about the water structure is that, of course, water is a liquid. It can move around and rearrange itself easily with minimal disruption to the number of hydrogen bonds between water molecules. And so it could easily solvate those groups which would be otherwise abandoned when the base pairs broke apart. So that leaves us in a situation where it's not at all clear that the hydrogen bonding between the bases actually contributes very much, if anything, to the stability of the double helix, because to a crude approximation, we're just replacing one set of hydrogen bonds with a different set of hydrogen bonds. Now, we should mention one piece of evidence which is often cited in support of the idea that these hydrogen bonds within the base pairs stabilize the structure, and that is that different segments of DNA vary in the relative amount of GC base pairs compared to AT base pairs. And it's been known for a long time that the higher the content of GC, in other words, the more of these there are relative to these ones, the more stable the double helical structure is. So the higher temperature, for example, you need to go to make it unfold, the more of these you have. And so people pointed at that and said, well, that's because you have to break 3 hydrogen bonds when you pull this pair apart, but you only have to break 2 to break this pair apart. Now, what we're now saying is, yeah, but that doesn't matter because you replace them with hydrogen bonds to water. So how do we get around this difference in the stability? Well, what we have to consider is the classic point that just because there's a correlation between two things, it doesn't mean that there's a causal link. In other words, yes, it's true that there are more hydrogen bonds between the bases here than there are here, and it's also true that the more of these you have, the higher the stability of the double helix. But that doesn't prove that it's the extra hydrogen bonds that create the extra stability. So what we at least have to leave open is the possibility that there could be other factors which make these stabilize the structure more than these do, and the hydrogen bonding is just coincidental. So once we've examined what the other stabilizing factors might be, we can examine the possibility that there could be another explanation for the effect of the higher GC content stabilizing the structure. Now, of course, it's not really quite that simple, and people do still argue a great deal about whether and how much these hydrogen bonds within the base pairs may contribute to stabilizing the double helix. For example, we've obviously oversimplified by saying that a hydrogen bond is just a hydrogen bond is a hydrogen bond. They're going to differ in the strength of individual hydrogen bonds depending on their geometry and their length and so on. And there's also a whole issue to do with the entropy of the water. In other words, what effect does the change in the distribution of hydrogen bonds between the water and the DNA, what effect does that have on how disordered the water particles are, and therefore on how dispersed their energy is? For all these reasons, it's not a completely clear-cut picture, but I think it is fairly compelling that at most these hydrogen bonds make a minor contribution to the stability of the helix, because all you're doing when you form them is replacing one set of hydrogen bonds with a different set. Now, the big issue that then raises is that, of course, these hydrogen bonds are said to be the key to the function of DNA as a repository of genetic information. Because the idea is that when DNA is copied, if you have a guanine, for example, in one strand, when you make a complementary strand, when you're synthesizing a new complementary strand, it has to put a cytosine in the place opposite so that these hydrogen bonds will form. And if there's a thymine, then it has to place an adenine opposite it so that it can form these hydrogen bonds. And the simple picture was, well, because otherwise the double helix won't be stable. But what we're now saying is, yeah, but actually these don't really make much contribution to the stability of the double helix. So is that really true? And that's something we need to examine, because if we're questioning the whole basis of the complementarity of strands, then that's a pretty big deal. To examine that question, let's come back to the structure and remind ourselves that it's not really full of all this empty space that you seem to be able to see when you draw it in this kind of way. If we change it to a more realistic representation of the core, as we've seen before, there is no empty space there. So the core, where the bases stack on top of one another, is extremely densely packed, and there is absolutely no room for any water inside the core. So all those waters that we can see around the DNA are outside here. None of them are inside there. And that's very important, because what it means is that if those hydrogen bonds can't be formed inside the core, then they can't be replaced with hydrogen bonds to water. So here are the Watson-Crick base pairs again. So GC with its 3 hydrogen bonds there between complementary groups, and AT with its pair of hydrogen bonds. And let's have a look at what happens if we try to mismatch the bases. So let's bring in a thymine. So here's a thymine coming in, and let's see what happens if we try to put it where the cytosine goes. And you can see the problem, that if we put it in the same position, we do actually manage to get one hydrogen bond across here, because we've got the NH delta plus and the O delta minus, so that's fine. Here, we've just got no hydrogen bond. So we've got the 2 delta negative carbonyl groups repelling one another and nothing in between. And here, we've got the 2 NHs actually clashing. So those are really not going to fit together properly like that. And so that means that if we did form a pair like that inside that core structure, first of all, we would be missing out on 2 hydrogen bonds that should be there. And those hydrogen bonds would be there between these bases and water in the unfolded state. So we would effectively be losing 2 hydrogen bonds. And that's going to make it unfavourable to include that base pair in the standard double helical structure. Added to which, we're going to get some repulsive energy, particularly from this clash of the NHs there. We can do the same thing if we try to bring a cytosine in to pair with adenine. So again, here comes cytosine. If we try to put it where the adenine should go, you can see this time it really is even worse, because we've got a clash of NHs there, and we've got no hydrogen bond at this position or at this position. Possibly you could get a water in there, so you might be able to deal with that one. But here and here in the core, got no hydrogen bonds, but those hydrogen bonds would be there in the unfolded state, being formed between the bases and water. And so this is going to be very unfavourable, because you're losing that hydrogen bond enthalpy and getting some repulsive interactions across there. Okay, so you can probably understand this better with the help of a soup-based analogy. So here is a bowl of delicious soup, and I have a strong driving force to eat this soup because it's nutritious and delicious, as you can see. However, this might happen. Now suddenly, even though I still like soup just as much as I did before, I feel disinclined to eat the rest of this soup because it's now liberally mixed with Labrador saliva. So the point here is that the driving force for eating soup is still there. The driving force isn't that I like soup because it isn't mixed with Labrador saliva. That's not what the driving force is. That's just a necessary condition that has to be met. The driving force is that I like soup, but unless the other necessary condition is met, which is that it's not mixed with Labrador saliva, then it's still not going to happen. And that's exactly the case with the DNA, that it will fold up, and the driving force is not the formation of those hydrogen bonds in the core. The driving force is something else which we've still got to discover, but unless that condition is met, unless those hydrogen bonds are formed, then it's not going to happen. And then you can have the rest of the soup. We're left then with the idea that in the context of the double helix structure, the bases are attached to the sugars at these specific positions, which are dictated in their position relative to one another by the architecture of the backbone as it folds into the double helix. And we've seen that if we keep these points more or less fixed, that if we try to bring in a different pairing, as we already saw, if we tried to bring thymine in, for example, it didn't work because we couldn't make complementary hydrogen bonds. However, if we relax that condition that requires these points of attachment to be in exactly the same place, that situation's not quite so clear-cut. So here is the original position, but if we allow this to slide up a little bit, we can do that. Now that's very interesting, because what we now have is a guanine-thymine base pair, and you can see there is a hydrogen bond that can form across here, and another one that can form there. So GT is a viable base pairing if we have the freedom to change the relative positions of these points of attachment. What about pairing adenine with cytosine? Well, remember we saw before that if we try to do that, it just doesn't work at all. If we try to put them in the corresponding places, we get no complementary hydrogen bonds forming. This time, sliding it up and down doesn't really help very much. We could form one hydrogen bond if we do that, or one hydrogen bond if we do that, but we can't get more than one. Although, you know, if you take the view that replacing them with hydrogen bonds to water is just as good, that may not matter. But, of course, these structures, in the absence of stabilizing non-covalent interactions, are very flexible. We can rotate around single bonds and things, and we could imagine that if the chain reorganized itself, we could flip this cytosine so that it was upside down. So we can do that. So here we have a cytosine which is coming in from the opposite angle, and you can see that if you do that, you can now form a pair of hydrogen bonds across here and here. So again, more possibilities, but dependent on changing the points of attachment to the backbone. So what does that mean? Well, what it means is that the specificity of base pairing depends on the whole helix being stabilized by whatever the stabilizing force is, and the idea that other arrangements of the backbone will be less stable because they won't be as compatible with that stabilizing force. So the complementarity of hydrogen bonding being the basis of the transfer of genetic information, we're not questioning that, but we're saying that it depends on that hydrogen bonding being a necessary condition for forming a stable core structure, and that that stable core structure is defined by the architecture of the double helix, and that must be stabilized by something else. So where we need to go next is to really establish what's so special about that particular helical arrangement that makes it stable and defines these base pairs.